Tuning into this poster, which will cover a signaling network model that we built to predict the effects of FUS on blood-brain barrier protein expression in brain endothelial cells. As a quick review, the blood-brain barrier refers to the largely impermeable vascular network throughout the brain. In contrast to capillaries and other tissues, blood vessels in the brain prevent the transport of most molecules from the bloodstream into the sensitive brain parenchyma. While this barrier is crucial for the health of the brain in most settings, it can also inhibit delivery of therapeutics in a disease context. One of the most important features of the BBB is the junctions between endothelial cells, which serve to create a tight seal and prevent passage of most molecules into the brain. These junctions consist of occludins, claudins, jams, and cadherins, among others. We already know that focused ultrasound can be used in conjunction with microbubbles to transiently disrupt these proteins and open the BBB. But what we wanted to investigate here is the longer-term impact of FUS-mediated transcriptional changes on the BBB and how those might work in concert or antagonistically with gene therapies of interest. This is just a schematic of the logic-based ordinary differential equation-driven signaling network model. So it consists of 51 molecular species, and those are connected by Boolean logic gates, including and, or, and not gates. The inputs for our system were the desired gene therapy payloads. So we looked at growth factors including VEGF, BDNF, and NGF. Both BDNF and NGF begin in a protoform, which can either be cleaved by an enzyme like plasmin or furin or MMPs into a mature form of those growth factors, or they can join a co-receptor called sortilin to activate a different downstream receptor. As for outputs, we looked at a number of different outputs for the system, including those BBB proteins of interest, occludin, claudin 5, and VE cadherin, as well as a number of different transcription factors, including beta-catenin, HIF1-alpha, and CREB, and an apoptosis phenotype. In order to simulate the effects of focused ultrasound in the model, we looked at different transcriptomic signatures of endothelial cells following treatments of the brains of mice with focused ultrasound. So we used FUS to target the right striatum, sorted FUS transfected cells based on a delivery of a fluorescent transgene, used single-cell RNA sequencing to identify the endothelial cell population, and then looked at differential transcriptomes between that endothelial population and a control population. And one transcript in particular that was significantly upregulated in the FUS-treated endothelial cells was cathepsin D. We were able to incorporate cathepsin D into the model with relative ease because it's been shown to upregulate both AKT and ERK, which are already existing species in the model. So we therefore simulated the effects of FUS on the endothelial cell network as this bolus input of the cathepsin D node. We conducted knockouts of each node to do sensitivity analysis of the network and found that the VEGF pathway was particularly influential on the network at large, while nitric oxide signaling is particularly sensitive to many different node knockouts. When we looked at the impact on the blood-brain barrier in particular of different knockouts, we found, which we did by assessing the cumulative effects on claudin 5, occludin, and VE cadherin nodes, we found that VEGF and PLC gamma signaling play the largest role in affecting BBB integrity, as the model currently stands. When we added the FUS-induced cathepsin D upregulation into the model, we found that the transcriptional changes result in blood-brain barrier disruption, as shown by this decrease in claudin 5 and occludin expression, a decrease in apoptotic signaling, as well as increased production of the various transcription factors present in the model, which could have a wide range of downstream effects that we hope to capture in the next iteration of the model. So, in conclusion, we've demonstrated that computational models can simulate the cell type specific effects of FUS, and that low pressure FUS is predicted to decrease the BBB integrity based on decreased expression of tight junctional proteins in endothelial cells. It's also predicted to decrease apoptosis and increase transcription factor expression in these brain endothelial cells. And all of these findings may help us predict the effects of different gene therapy approaches in the long term using FUS-mediated delivery. I'd like to thank my lab, our collaborators, and our funding sources, and thank you so much for listening.